Dear learners, I welcome you all to attend this session. Today, I will discuss about transmission electron microscope. So, these are the learning outcomes. After attending this video lecture, you will be able to understand the basic principle of transmission electron microscope. You will differentiate the SEM and TEM in detail, and you will explain applications, advantages and limitations of transmission electron microscope. So it will be very clear after attending this lecture. You will get the very clear difference in detail about SEM and TEM. So dear learners, I will start with the lecture. So first of all, what is transmission electron microscope? So basic principle over here is transmission, and it gives the measurement about inner structure of molecule or inner structure of material as the scanning electron microscope. As I discussed, in a scanning electron microscope, it gives the information about outer structure, but this transmission electron microscope will give information about the inner structure. Now here the design. This is the design of a transmission electron microscope and a light microscope. So if we compare the design, it is very clear. Both are similar, but it's substituted. This transmission electron microscope is substituted in some parts. Over here in a light microscope, there is a lamp, means there is a light, but it is substituted over here by the electrons. Then if we move towards the glass lens, then it becomes electromagnetic lens. Then here this is the specimen. So here we will put the sample on the slide, but here we have to put the sample in a TEM grid. There is a grid specifically made up for TEM. That's why it is known as TEM grid, because here we require a very thin sample. You can say ultra thin sample, and that's why we have to use this specifically made TEM grid. Now here again the same. Now in place of eye, here there is the replacement of a phosphorescent screen, or you can say there is a fluorescent screen. Otherwise here also the camera is there. It's a CCD camera where the picture, where the image will capture. So this is how it is different by the design. Now we will see the difference between TEM and SEM. Dear learners, I want to clear the difference first before starting the principle of TEM, because we know about SEM, so we should know how it is different. Then I will start the principle. So first of all, I outline some basic difference. As I discussed, SEM will give the outer structure and TEM will give the inner structure. So this is the point over here. The image is a two-dimensional projection of a sample, means it will give the inner structure, but here the image is of a surface of the sample, so it will give the outer structure. Now here the electron beam passes through a thin sample, but here there is no limitation for thickness and it is passes over the surface of the sample. Here the specifically prepared thin samples are supported on a TEM grid, as we have seen in the design, but here it can be of any thickness and it is mounted on an aluminium stub, or you can say it will be mounted on the specimen chamber. Now here specimen stage halfway down the column, but here it is in the chamber at the bottom of the column. And here the image shown on the fluorescent screen, but here there was a TV monitor. So this is the very basic difference about TEM and SEM. Now it will clear by this picture. Here this is the structure, this is the instrument of a TEM, this is the instrument of a SEM. So we can see the clear difference. Here there is a, as we have seen, the electron beam is there, it passes over the surface and will get the image like this. It's an outer structure, but here the specimen is there, it's a very thin specimen you can say, and it is passed like this, and we will identify the inner structure. Now we will move toward the principle. So here also there is a comparison between SEM and TEM. So dear learners, in SEM, as I have discussed, there are three types of electrodes. The principle remains same, but here there was scattering, here there will be a transmission. The basic principle of TEM is this T means transmission. Now what is the principle? So here we can see by the design, then we will come again over the slide. See here what happened. Of electron is transmitted, and with the ultra thin specimen over here, as it interact, as it passes, so we are getting the image, as the interaction of electron transmitted through the specimen, and the image is magnified and focused onto this fluorescent screen and on the layer of the photographic film, and it is detected by one sensor, and that you can say the camera, that is CCD camera. So it is the basic principle, there is a transmission. Now see, as in SEM, what we have seen, there are backscatter electrons, secondary electrons as well as X-rays. But in TEM, there will be generally a scattered electron and unscattered electrons. But mainly we have to focus on two types of scattered electron. It's elastically scattered electrons and inelastically scattered electrons. Now we will see what is this type of electrons. So as we know about backscatter electron, this is no point of discussion. What is elastically scattered and inelastically scattered electrons? So elastically means an electron of a primary beam is scattered by the electrostatic interaction with the positively charged nucleus of an atom, means you can say your specimen, an angle of less than 90 degree. It is scattered at the angle of less than 90 degree and it is known as elastically scattered electron. What is backscatter? It's scattered more than 90 degree. And here inelastically scattered electron means electrons can also lose energy while interacting with electron cloud. Means you can say while your electron beam, while your primary electron coming towards, and it will do interaction with your specimen, and whatever the electron cloud of specimen is there, it will lose the energy, and this kind of electron called inelastically scattered electrons. Now see, this is the very clear picture of electron specimen interactions. See, this is our primary electrons, means initial energy, it's a E0. Now this is our nucleus, means you can say sample, specimen. So when this primary electron come over here, it will be backscattered. The E is equal to E0. Whatever energy it have, it's equal to E0. Now when it will be scattered, see this is a scattered electron, but again it's divided into two parts, elastically scattered electron and inelastic electrode. So see what is elastically. Here the angle is less than 90 degree. This is the electron cloud. Now here there is a higher angle and E is equal to E0. But in inelastic, what happened? The electrons lose energy over here and it will be at a low angle. So this is the energy difference over here, and this is indicate the unscattered electron. So now this is the clear difference between scattered, backscattered, or you can say elastically and inelastically scattered electrons. So in biological analysis by TAM, the elastically scattered electrons are mainly used for imaging, and the inelastically scattered electrons will be recorded as with the image device. We'll see the advantages of transmission electron microscope. So it will produce a high resolution and magnification. As we know, it produces high resolution. It gives information of elements and compounds. Obviously, it will give us the inner structure. So it will give us the information about the inner structure of elements and compounds, and detailed image obtained. Because here we are getting whatever image we are getting, it's on a fluorescent screen, and we are using this CCD camera as a sensor. So these are the advantage of transmission electron microscope. Now we will move towards applications of transmission electron microscope. So this is the most important. First is virology, means we know how it is used for investigation of virus structure. We can measure the inner structure of virus by using transmission electron microscope. Then here it is. Second is cellular tomography. So what is this tomography? Dear learners, tomography means to imaging by sectioning through the use of any kind of penetrating wave. You can say it is used to obtain a detailed 3D structure of a subcellular macromolecular objects. So it is a very important application. Then it is used in a cancer research. Here we can study the ultrastructure of a tumor cell. Then it is used in a toxicology. What is toxicology over here? So it is used to study the impacts, or you can say the effect of environmental pollution on the different levels of a biological organization, and it's used in a silicon and computer chips. Means we can use it as a production of the silicon chip, or you can say manufacturing of the computer chips. So it have also the wide application like scanning electron microscope. Now see, it have some limitations. Why limitations? Because, or what is the limitations? So samples are limited to those that are electron transparent. Samples are limited to those that are electron transparent. It is first limitation, because it's a transmission electron microscope. Then it's difficult to handle. Now why it is difficult to handle? Because here there is a tricky sample preparation, as it requires a very less sample, or you can say a very thin sample. It's have a very tricky sample preparation. So these are the limitations. Thank you. If you have any doubt, you can contact me on this email ID, or you can WhatsApp me on this number, or you can call me on this number. We will see the instrumentation part of transmission electron microscope in my next lecture. Thank you.